You almost know.
All right, good evening, everyone. We'll go ahead and get started. Wanted to wait a couple minutes to let everybody get into the room and get situated. My name is Jeff Warren. I'm the Executive Director of the North Carolina Policy Collaboratory. We are pleased that you are joining us this evening to spend a few hours with a handful of our researchers from the PFAS Testing Network. And this team that we've had, and we've just turned in our final report to the legislature after about a 2-year intensive statewide sampling effort across the state. Before we get started with our panelists, I first want to introduce 2 representatives from the General Assembly and allow them a chance to give some introductory comments. We've got Representative John Soka and Senator Kirk Dewitt. We'll start with Representative Soka.
Thank you very much, Jeff. I just want to tell all the participants here that this whole briefing tonight is as a result of your questions that were brought up at a local meeting held out in the Ruritan building out in Grays Creek. I know, I'm well aware that not everything the state has done, either DEQ or maybe myself or Senator Dewitt or government in general, has been well received. But I will assure you that the people who represent you, which is myself and Senator Dewitt, are doing everything we can. And more importantly, we're actually listening to what you're saying. The whole issue of how does Gen X affect large animals and small animals, that was your idea. It wasn't our idea. We moved forward. We got the funding to do that, and we'll hear the results of that study tonight. So I know that this problem is persistent. I know that it's taken a long time to get things done. We continue to work legislatively to get relief out to the Grays Creek area. And there's a number of things that we have done. I just want to say that please don't give up. And when you have ideas like this idea about the effects on animals and crops and other things that now are being studied, continue to tell us of those, and we'll do everything we can to get those studied. So again, thank you for attending tonight.
Thank you, Representative Soka, representing Cumberland County. Senator Dewitt from the other chamber and the other side of the aisle, also Cumberland County.
Thanks, Jeff. I appreciate being here tonight and the opportunity to say a couple comments. Thanks for all that your team is doing. As Representative Soka said, this is really because of the community and what we heard from the community and what we're able to look to do to get this research done. I just want to point out, and Jeff said it, I am from the other side of the aisle as Representative Soka, and, water this is a bipartisan issue. This should not be a partisan issue. And I think that's the approach that Representative Soka and I have taken and will continue to take. because we need to continue this research. We need to maybe even look at additional research after we come out of the results from tonight. And I think we're committed to doing that. Well, I know we're committed to doing that. He and I will continue to listen to what's happening in our community and try to be as responsive as we can, as responsive as government can be, to what we hear from the community. So again, thank you for being here tonight. Thank you for the research that's been done, and know that you have a delegation that's committed to finding solutions to what we have happening in our community. So thank you so much.
Thank you, Senator. And thank you to both of you. We appreciate it. Two of our legislators joining us this evening and for helping to facilitate our ability to share some of the latest research with their constituents and people from all across the state of North Carolina and beyond. At this point, we'd like to take a quick poll so that we can have a sense of where our attendees are joining us from tonight. So if you would, please take a second to participate when that poll pops up, and there it is. We'll do this, we'll leave this open for another 15, 20 seconds or so. All right, I feel with our General Assembly members here, I need to say clerk will lock the machine and record the vote. All right, so that's great. 21% from outside of North Carolina. So there is a great interest in PFAS across the country and across the world for sure. We have a packed agenda tonight, including 2 Q&A sessions. We will be monitoring the Q&A during the presentations, so feel free to use that function throughout the evening. And there's a Q&A button down at the bottom of your Zoom screen. At this point, I'm going to turn it over to our researchers. The first to go will be Professor Detlef Knappa from NC State University. So the floor is yours, Detlef.
Thank you, Jeff. Trying to set my screen up here. Okay. So good evening, everyone, and thank you for attending tonight's forum. So I want to start out by introducing the topic of PFAS, or per and polyfluoroalkyl substances, here in North Carolina. PFASs are a class of man-made chemicals. There are many subclasses and thousands of individual compounds. Some of these have been studied for a long time, like PFOA and PFOS, for example. But there are also many PFASs about which we know very little or perhaps nothing. And we really began this journey here in North Carolina also about 5 or 6 years ago with the discovery of a number of PFASs for which essentially no information existed. How do PFASs get into the environment? PFASs are released into the environment by fluorochemical manufacturers, but then these fluorochemicals are also used in a lot of products, including many consumer products. And so during the production, use, and disposal of these products, PFASs also get into the environment. So if you'd like to learn a little bit more about products in which PFASs commonly are found, you can go to this web link here at the bottom of the page, the 6classes.org website. So in North Carolina, historically, in my research group, we have focused a lot on the Cape Fear River Basin. And the Cape Fear River Basin is the largest watershed in the state of North Carolina. And it supplies drinking water for about 1.5 million North Carolinians. About 1 million of those North Carolinians are impacted by industrial wastewater discharges that add to the water PFASs and other unregulated contaminants. We have in the Cape Fear River watershed a PFAS manufacturer, used to be a DuPont facility, now Chemours in the central part of the basin. But I'll start the journey up here in Pittsboro, and I'll also talk, of course, about Wilmington, North Carolina. So when we look at PFAS occurrence in Pittsboro, the first data set was published in 2007 from samples that were collected in 2006 by US EPA researchers Mark Striner and Andy Lindstrom. And the blue data sets, these bars show PFAS levels, individual PFASs that are listed here at the bottom of the graph. So in 2006, the tallest bar was PFOA. That was the compound, the PFAS compound that occurred at the highest levels in Pittsboro. When we then fast forward 12 years to 2018, the speciation of the PFAS has shifted. So the dominant species now is this PFHXA. So we shifted from somewhat longer chain PFASs to somewhat shorter chain PFASs during this time period. But as you can see here at the top, the sum concentration of PFASs actually was somewhat higher in 2018 than it was in 2006. And these levels here are very high when we think about current drinking water standards that are being developed in many states around the country. For example, in the state of Michigan, some of the individual PFASs are now regulated at single-digit nanogram per liter levels, so less than 10 nanograms per liter. In terms of sources for PFAS in the Haw River and in the Pittsboro drinking water, it's upstream wastewater discharges, land-applied biosolids from wastewater treatment plants, landfill leachate that enters wastewater treatment plants, and also firefighting foam from at least one of the airports located upstream. So if we go down to Wilmington, North Carolina, we went to a 2015 sample a couple of years ago to learn what people were drinking in Wilmington at that time. And if we look at the traditionally studied perfluoroalkyl carboxylic acids, there was 120 nanograms per liter of them. The perfluorosulfonic acids, about 45 nanograms per liter. But then this was the big unknown that was not discovered by the standard methods that were in place at the time. There were many fluoroethers in the water, and this particular sample had around 130,000 nanograms per liter PFAS. And what I want to point out here is that Gen X in the end became the famous PFAS in the story, but the Gen X slice here is very, very small. There were many other PFASs in this water that were present at much higher concentrations. And as we learned from some of the disclosures that were made by Chemours, who discharges these chemicals or discharge these chemicals into the river through their wastewater, these compounds likely began being discharged into the Cape Fear River since 1980. So if we go to this cartoon here of a manufacturing facility, the wastewater that was discharged into the river impacted Wilmington and Brunswick County and Pender County. But the facility also emitted PFASs into the air, and these PFASs were then deposited through dry and wet deposition back to the soil. Some of the PFASs get taken up by plants. Others will make it down into the subsurface and then contaminate the groundwater and private wells in this region. So for a long time, it probably also rained Gen X and other PFASs onto the properties surrounding the Chemours and DuPont, earlier DuPont facility in Cumberland County, Bladen County, and also in Robeson County. And here you can see some DEQ data showing Gen X levels in rainwater And you can see these Gen X levels were elevated even 5 miles away from the plant. So the net result of this air emission and then deposition of PFASs was private well contamination. And you can see in this recent map here that over 5,000 private well samples have now been taken, and many of the wells, over 4,000, are contaminated with PFASs to some extent. This is an area that is very large and extends now well beyond 10 miles away from the plant. Apart from the fluorochemical manufacturing facility in the central part of the Cape Fear River Basin, we do have other PFAS sources. So for example, here I'm showing the Greensboro Airport, and when you look at a creek that drains the airport, there are high PFAS levels in this creek. And this creek then runs into Lake Brand, which is one of the drinking water sources for the city of Greensboro. So that is related to firefighting foam at this particular airport. And finally, land application of biosolids can be an important source of PFASs. So here I'm showing Cane Creek Reservoir, just one of the drinking water sources for Chapel Hill Carrboro. And just upstream from this reservoir are land application sites for biosolids. And these are some US EPA data that show that PFOS and PFOA levels in the creeks that drained these fields were highly elevated and are now impacting Cane Creek Reservoir. So as a result of these many sources of PFAS inputs into the environment, people have many questions about PFASs. For example, are PFASs in my drinking water? Are PFASs in my drinking water that standard methods do not detect? Are PFAS in me, and at what levels, and what are the health effects? Also, are PFASs in my food? For example, the fish I catch or the food I grow in my garden. And then also, how can I get PFAS out of my water? And so these community questions really provided the basis and also for the research that we have been conducting as part of the PFAS Testing Network. So with that, I'll turn it back over to you, Jeff.
Thank you, Detlef. That was a great overview and introduction into PFAS in general and PFAS in North Carolina. As you know, in 2018, the General Assembly appropriated just over $5 million to the Collaboratory at UNC-Chapel Hill to run a statewide PFAS project, and we did so with 7 different university campus partners. So the next person you're going to hear from is Dr. Wanda Bodnar. Sorry for stumbling over that, Wanda, who's a faculty member at the Gillings Global School of Public Health at UNC-Chapel Hill. Wanda did an amazing job pulling all of these data together in the final report, which was just delivered and is available at the Collaboratory's website, as well as the PFAS Testing Network website, as really the linchpin of the project management team. So I'd like to turn it over to you, Dr. Bodnar.
Thank you, Dr. Warren. You actually took my first sentence, so I'm a little bit thrown off, but we'll keep going. Thanks everyone for joining us. It's my pleasure to give a broad overview and introduction into the PFAS Testing Network, our members, our organization, and our research objectives. So shown here, I have the organizational chart for the PFAS Testing Network. As Jeff said, as part of the 2018-19 state budget, the North Carolina General Assembly gave a just over $5 million appropriation to the Policy Collaboratory to assemble this network of academic researchers, including faculty, scientists, and students from 7 North Carolina universities. And we were charged with collecting very important data that's needed by our state regulatory agencies and lawmakers to establish policies and develop guidelines to protect human health, wildlife, and the environment. And seen here in green is our management team, the program director, Professor Jason Surratt at UNC-Chapel Hill. I'm the scientific program analyst, and Manal Khan is the program assistant. So the three of us form the management team. We also have executive advisory committee members who are experts in PFAS research and have been working in the PFAS area for a number of years. In blue are highlighted our research teams. We actually have 10 research teams. Team 1 is co-led by Lee Ferguson and Detlef Knappe, and May Sun from UNC Charlotte is also an important part of that team. Team 2 is our private well risk modeling team led by Dave Genero at NC State and Jackie McDonald Gibson, who's now at Indiana University. Team 3 is our Pfas Removal performance testing team, led by Professor Orlando Cornell at Chapel Hill and May Sun at UNC Charlotte. Team 4 is our air emissions and atmospheric deposition team, led by Ralph Mead in UNC Wilmington and Barb Turpin, UNC at Chapel Hill. And then Team 5 actually consists of 6 teams. It's co-led by Jamie Dewitt at ECU and Rebecca Fry at UNC-Chapel Hill, and they have an additional 4 team leaders that I'll mention in the next slide. And then shown here in yellow are our research support teams. We have Team 6, which is our communications team. They are very critical to all the work that we do. They interact with the public, with our stakeholders. They provide training to our investigators to help us present information in a way that's accessible and understandable to the public. That team is led by Kathleen Gray at Chapel Hill, Jane Hoppin at NC State, and Jory Weintraub at Duke. Our Team 7 is our data science and management team, led by Chris Lenhart at Chapel Hill and Helena Mitsova at NC State. They're responsible for taking all of the findings, all of the results that we generate and putting those into a database, into a data hub portal that you'll be able to access to view the data in a number of different visual ways. And then we also we're very fortunate to have a synthetic organic chemist in the network who has been busy trying to develop new synthetic strategies for PFAS analytical standards that you can't buy commercially at this time. So let me tell you a little bit about each of the research teams. Our Team 1 statewide baseline water testing team, their objective is to measure PFAS levels by targeted analysis at surface water intakes and groundwater wells across the state. And they are also running at UNC Charlotte a method to estimate the total organic fluorine in the samples. And then they can compare that with the summed levels of PFAS from the targeted analysis and determine how much of the perfluorinated compounds we're accounting for. We also have non-target analysis ongoing to identify more PFAS. So beyond the 48 to 50 PFAS that we analyze in our targeted analysis, we also do suspect and non-target analysis to identify new PFAS that perhaps are emerging that we weren't aware of before. Our private well contamination risk modeling team, they're trying to understand how PFAS move through groundwater and why some private wells are contaminated while others are not. So they're developing computer models that incorporate data like well features, landscape and geology, location, in order to predict the risk of well contamination with high levels of Gen X and other PFAS. They're doing calculations to figure out how long it'll take for PFAS to flush out of the aquifer just by natural groundwater flow. And they've actually produced a user-friendly web app that private well owners can use to predict their risk of well contamination. Our PFAS removal performance testing team is evaluating commercial filter options for removing PFAS. These are things like granular activated carbon, ion exchange resins, high pressure membranes like nanofiltration membranes and reverse osmosis membranes. They're also they tested tap water with in-home filtration options. So either whole home filters or point of use filters under the sink or refrigerator, or pitcher filters like the Brita filters that you have on your countertop. They're also developing new materials. So a combination of fluorine containing and ion exchange containing resins. And you're going to hear some more about that later on. And as well as a method for PFAS destruction. So once you've generated all of these PFAS rich waste streams, we need a way to deal with that. And so May Sun's lab at UNC Charlotte is working on an electrochemical degradation method where they can mineralize and destroy the PFAS. Our air emissions and atmospheric deposition team, they are looking to identify PFAS in air and rainwater. So they're looking at gas phase PFAS as well as particulate fine matter, or also called PM 2.5. They're looking at wet rain and snow deposition as well as dry deposition. They're also analyzing the geographic distribution of PFAS. They have sampling sites across the state. And so they're trying to figure out sources by measuring different PFAS profiles at different locations. And they're also doing some laboratory experiments investigating multi-phase atmospheric chemistry. That's a wide open area of research looking at how PFAS and PFAS precursors get transformed by atmospheric chemical processes, and what are the products from that. Then our applied research projects. So this is the team led by Jamie Dewitt and Rebecca Fry, and we have 6 different research areas within this umbrella team. They're looking, the first team, which is led by Mort Barles at NC State, is working on wastewater treatment plants, landfills, and those kinds of sources of PFAS to surface and groundwaters. So he's looking at the importance of municipal solid waste landfills, construction and demolition debris landfills, domestic wastewater and biosolids, and how those are contributing PFAS into our environment. Scott Belcher, who you're going to hear from later, also at NC State, is studying PFAS bioaccumulation and biomagnification in ecologically relevant species. So looking at different species of fish and alligators, determining the presence of PFAS, which PFAS at which levels, and their data is going to help determine whether or not consuming fish caught in the Cape Fear River basin increases human exposure and bioaccumulation of PFAS. Then we have Jamie Dewitt at ECU is looking at PFAS immunotoxicological response using a mouse model. She's studying the impact of emerging PFAS on the immune system and comparing that to what we know about legacy PFAS like PFOA and PFAS. She's looking at effects on the body, the organ weights, immune cell populations, antibody response, functional responses. And her work has been absolutely critical to informing regulators and policymakers about potential health impacts of PFAS. And then for the crops, Owen Duckworth at NC State, which you'll hear from him as well, he's looking at the, PFAS uptake in food crops and the effects on soil composition on that uptake. So as you change the different percentage of compost, how does that affect PFAS, and where is PFAS actually going within the plant as it's taken up? And so that work should inform management practices to help reduce PFAS exposure through consumption of crops. Rebecca Fry at UNC-Chapel Hill is studying the effects of PFAS on pregnancy and placental health. So that's an area of expertise in her lab. She's doing both human population studies, looking at a pregnancy cohort, analyzing PFAS in their drinking water and in their actual samples, placental samples, And they also are doing laboratory studies looking at gene expression and pathways that are involved once exposed to PFAS. And then the last team is led by Nick Luke at NCA&T, and he's actually a mathematician, and he's working with all of our teams using their data to develop computer models to predict where PFAS go in organisms and the environment. He also works on reference dose and benchmark dose modeling to try to figure out what might be protective levels of PFAS in human exposure. There's one other project that's not captured here. It's a project that I actually led, which came in a separate legislation, collecting a statewide inventory of firefighting foams, so the aqueous film forming foams, or AFFF, across the state. And so that's another project that I'd be happy to answer questions about. So here is how you can access all of this wonderful information. I have the link here to our website, the ncpfasnetwork.com, and I also have our email address, which is hosted at UNC, and myself and Manal Khan and the management team monitor that, and we can forward any questions that you send via that email address to the appropriate investigators to get more information. So if you go to our website, and you hover over resources, you'll see a black box pop up with graphics and printed materials and newsletters. If you actually click on resources, you'll be able to access all of our former quarterly progress reports. And then under data and tools, if you hover over that, you'll see a link to the data hub, which should be active sometime this month. And you'll be able to access all of the data and visualization tools, as well as the GenX predictive tool for the private well owners. If you click on data and tools, you'll actually be taken to a listing of the water, baseline water testing results. So all of those reports are available there as well. And then if you click on the final report, if you hover over it, you can get the black box with the AFFF inventory project report. So that's separate from the PFAS network report. But if you just click on report, it brings up this page, and you can click on the full final report link, and that will bring up I believe in a separate window, a 95-page report that was submitted to the General Assembly on April 15th and subsequently revised and resubmitted on April 30th, just to correct a few minor things. We have the executive summary and recommendations and some supplemental materials here as well, including a description of the chemical synthesis that has been performed in the last couple of years. And I think that's all I had. I want to acknowledge that the PFAS Testing Network is over 110 people. And it's just an incredible amount of work, a great team of people to work with. And if you have any questions at all, anyone here would be happy to answer them. Thank you.
Thank you, Wanda. Thank you very much. Y'all have had a huge effort, and you brought this all home after more than 2 years of work into a great final product. Moving on, at the other end of Tobacco Road from Duke University, the darker blue, Dr. Lee Ferguson is going to join us. He is 1/3 of the water sampling team. Detlef Knapper was the other third, and Dr. May's son at UNC Charlotte, he's not with us tonight, was also an important part of that. So Lee, I will turn it over to you.
Great, thank you, Jeff. So I'm just going to get my pointer set up here. Okay, great, thank you. Yes, so I'm going to be talking about the results from our statewide drinking water testing network and our statewide analysis, sampling and analysis. This is a collaboration between my laboratory at Duke, Detlef Knappe's laboratory at NC State, that we've already heard from, and May Sun's laboratory at UNC Charlotte. For this project, we were charged with sampling all municipal and county active water systems across the state of North Carolina to test for PFAS in raw drinking water. And so what you can see here is the map showing all of the sites that we've sampled. There were 376 total sample sites. divided among 166 surface water sites and 210 well water sources. And so what you can see from the map here is that the groundwater sources were really concentrated towards eastern North Carolina, and then a cluster here in western North Carolina, with central North Carolina and the Piedmont being primarily surface water sources of drinking water. And that's going to become important later as we talk about the results. So over the course of about a year and a half to 2 years, we've sent teams of researchers across the state to collect these raw drinking water source samples, bring back to the laboratory, and then these samples were analyzed using a newly developed targeted method that targeted 48 individual PFAS compounds that we could measure quantitatively down to about 1 part per trillion per compound. We also used a total organic fluorine method to look for total PFAS burdens in these waters, and we used non-targeted analysis to detect PFAS that might not have been captured during the targeted analysis. So just to jump into the results here, this is basically a graph showing the summary of our PFAS measurements in our public drinking water sources. And so the colors are broken down, as I'll show you in a moment, by compound class. And you can see basically the decreasing concentration as we go down the list of water stations. There are 376 sites on the y-axis here. The dashed line here is at the concentration of EPA's health advisory level of 70 parts per trillion for combined PFOS and PFOA. And if you zoom in on just the top section of this graph, what we'll see is the sample sites that had the highest concentrations of total PFAS, including the pink here, which are the perfluoroalkyl ether acids, as well as what we term the legacy PFAS, such as the perfluorocarboxylic acids and perfluorosulfonic acids here. The black names of water sites represent surface waters. The gray represent groundwater sources. And what we can see is that for 20 stations, 20 water systems had some PFAS greater than this EPA health advisory level set for PFOS and PFOA. 285 stations had PFAS concentrations greater than 10 parts per trillion. And the combined PFOS plus PFOA concentration only exceeded 70 parts per trillion in 2 stations. And that was the town of Maysville here and the Orange Water and Sewer Authority here. And so we'll talk about Maysville a little more later. Orange Water and Sewer Authority, actually Detlef already mentioned that. This seems to be impacted by runoff from biosolids applied to agricultural fields. But what we can also see is that 9 out of the top 10 most concentrated sites were dominated by perfluoroalkyl ether acids, and these are in the lower Cape Fear River. And if you look at the concentrations here, the total concentrations of PFAS at the highest concentration sites were close to or exceeded 400 parts per trillion. And if you compare this to recent work reported by the Environmental Working Group, you can see in that case, of course, the top concentration reported was actually from Brunswick County, North Carolina. But our measured PFAS concentrations, especially in raw drinking waters from the lower Cape Fear, were quite a bit higher than what we see across the country in drinking waters. If we dig a little bit more deeply into the occurrence of the various different compounds in the water samples, this is basically a summary of those 376 sites. The little gray pie charts here show the total detection frequency of the various different compounds. And what we can see is a cluster of these purple dots here with relatively high detection frequency, as well as these green dots here. PFOS and PFOA, together with the short chain carboxylic acid PFAS compounds, were the most frequently observed compounds in drinking water sources. These ether acid compounds and sulfonates were measured only east of the Chemours plant in Cape Fear River water. So only 2 legacy PFAS concentrations greater than 70 nanograms per liter were observed, and both of these were in Maysville, North Carolina. Those are here. That's perfluorohexane sulfonate and perfluorooctane sulfonate. If we take a look at the geographic distribution of our total summed PFAS concentrations across North Carolina, what we can see is that the higher concentrations are red, orange, and yellow. The lower concentrations are blue, purple, and green. And we see a trend of relatively low concentrations for PFAS in most raw ground or drinking water sources in eastern North Carolina and again in western North Carolina, with some exceptions. We can pick out here the outline of the Haw and Cape Fear rivers, dominated by short chain perfluorocarboxylic acids up in the upper reaches near the Haw River, and the higher concentrations dominated by, for example, perfluoromethoxyacetic acids and some of the other ether acid compounds in the lower Cape Fear. There's also some higher concentrations we found in the Neuse River as well. We can pick out a few high concentration hotspots, such as here, Maysville, North Carolina, as well as some of the lower Cape Fear. But then overall, the trend is that the Piedmont and central part of North Carolina had the higher concentrations of summed total PFAS. And this is because of the fact that central North Carolina relies on surface water for their drinking water sources. And in many cases, those surface waters are downstream of a discharge of municipal or industrial effluent, or they may be impacted by the application of biosolids to agricultural fields. Maysville, North Carolina was a special case here. This was actually a surprise. We observed relatively high concentrations of PFAS in the waters in Maysville. This was a groundwater well, a single well for the town. The signatures here, including fluorotelomere sulfonates, as well as perfluorohexane sulfonate and perfluorooctane sulfonate, were indicative of contamination with aqueous film-forming foam. And this was really the only instance of significant groundwater contamination that we observed, except for an unused Wrightsville Beach well that had been contaminated by injection of Cape Fear derived drinking water to the aquifer. When we reported this finding to Maysville, they immediately stopped using the well and switched over to Jones County water supply. We also noticed high temporal variability in several sites, and this was especially noticeable in rivers, and especially the Haw River at Pittsboro. So here we're actually seeing a comparison of the complete round 1 data and round 2, which is actually still being measured across the state for our analyses. Round 1 in April of 2019, we had measurable but relatively low concentrations of PFAS dominated by short chain perfluorocarboxylic acids. However, when we went back a few months later in September, we saw much higher concentrations, close to 800 parts per trillion total PFAS, dominated by the short chain perfluorocarboxylic acids. And this really highlights the importance of understanding sources of these PFAS in areas that may receive both changes in discharge due to changes in industrial operations, changes in flow of the river because of precipitation and baseline river flow, as well as differences in treatment that may happen in wastewater. So this is really one of our main takeaways, is that we need to monitor these types of flowing systems. So here are some take-home messages. I know these are relatively verbose, but I just want to hit some of the highlights while you read them. 376 water sources tested. Again, there were 20 of these that had summed PFAS concentrations greater than the 70 parts per trillion PFOS and PFOA health advisory level. Some of the most important PFAS sources that we find in drinking waters for North Carolina are the Fayetteville work site, Chemours, which we've heard quite a bit about. Also, aqueous film-forming foam used in firefighting applications, municipal wastewater discharge, which actually the PFAS in that wastewater is not coming from the plant, but coming from industries that discharge to those plants, as well as runoff from fields that receive biosolids. The PFAS molecular profiles in North Carolina really vary quite a bit among drinking water sources, and this highlights these complex mixtures. The concentrations in some of these drinking water sources, especially surface water sources, river sources, have a lot of variability. The absorbable organic fluorine concentrations we measured varied quite a bit. They were high in a relatively minor number of sources. And then when we did our non-targeted analysis, the dominant signals corresponded to PFAS that we were already quantifying through targeted analysis. And this suggested that the targeted analysis really captured most of the analytically viable PFAS. So here I'll just leave you with some recommendations of what's needed. We need continued monitoring, especially for temporal changes, as well as to look at private wells. We need action by industry to phase out non-essential uses of these materials, enhance water treatment, stewardship, and then action at the public policy level. And with that, I'll close.
Thank you, Dr. Ferguson. And last but not least on this block, before we do some Q&A, and then take a little break and let people get up and stretch their legs, we're going to hear from Dr. Jamie Dewitt, who's led our toxicology portion of the study from East Carolina University. And as Dr. Dewitt will tell you, she is no stranger to testimony, and she's been up on Capitol Hill multiple times as well in front of our legislature. There are not many of these compounds that we know a lot about toxicology-wise, but there are a lot more that we do thanks to the efforts of her and her team. So Dr. Dewitt.
Thank you, Jeff, and thank you, everybody. And let me say that testifying before Congress is, and let me turn on my camera first. Let me say testifying before Congress is pretty terrifying. I know some members of our community have done it as well. Some maybe even got to meet Mark Ruffalo. I was not so lucky because I was traveling out of the country. But I wanted to talk a little bit about some of the toxicological outcomes that we have been describing for some of the PFAS that are relevant here in Eastern North Carolina and perhaps throughout the rest of the state and even the rest of the country. Before I do that, let me remind you a little bit about some of our goals and objectives for addressing the toxicity of some of these understudied PFAS. We really wanted to focus in on the immune system because we understand that in people exposed to PFAS and in experimental animals exposed to PFAS, the immune system appears to be one of the more sensitive systems. The National Toxicology Program reviewed the literature on the immunotoxicity of PFOA and PFOS and determined that they were presumed to be immune hazards to humans. So we really wanted to ask whether or not some of the understudied compounds here in North Carolina also had the potential to affect the immune system. And we asked three main questions about the immune system. Two of our questions were functional in nature. In other words, we asked the immune system to do something and then measured the ability of the immune system to do that thing. One of the things we asked the immune system to do was to respond to an antigen. This is really similar to a vaccine response, which is probably a response that's on everybody's minds right now as we're hopefully leading out of the pandemic. We also asked questions about a group of innate immune cells called natural killer cells to identify and kill target cells. These cells are really important when it comes to surveying your body for cells that might have mutated and maybe risks for cancer. So we wanted to ask if innate immune cells were being impacted by PFAS. And then we also asked some questions about numbers of immune cells because the numbers of immune cells can be clues about other parts of the immune system that might be impacted. But throughout our studies, we also observed the animals to see if they were experiencing any generalized sickness. We asked questions about enzymes within their livers, which can be a hallmark of PFAS exposure. We asked questions about the size of their livers. And then we've also shared tissues with many other researchers who are looking at effects of these PFAS in the lungs, in the brain, and possibly even in the kidney. We've also evaluated levels of PFAS in the blood and in the urine. So we can start to get a little bit of a handle on how long some of these compounds might stay in the body so that we can get a very rough estimate of their half-lives. We have broken our research into two phases. The first phase we studied, and Detlef can pick on me later about the acronyms, but these are just the initial acronyms that we used. In our first study, we looked at the ether compounds. We looked at a C3, which is PFMOAA, and we looked at that because it was one of the most abundant compounds. But then we looked at some additional ethers with 4 carbons, 5 carbons, and 7 carbons. We have one of our papers published. It was recently accepted, and it was my postdoc's very first paper as a toxicologist, so it took us a little while to get it put together. But we include data in that paper on the 3 compounds that I have highlighted there, the PFMOAA, the PFMORPRA, and the PFMOBA. And I'll talk a little bit about some of those results. But what I wanted to emphasize is that when we start a project with new compounds, we often don't know how much to give test organisms. We certainly don't want to give so much that they experience death. We're interested in sublethal effects. So our first experiment with PFMOAA, we used doses that were quite low, but were really similar to what was being discovered in the water. We later found that this compound is very rapidly excreted from mice. So the amount that was built up in the mice was actually below limits of detection. So we didn't see any changes. We then increased the amounts in some subsequent studies with additional compounds, and we've also done a repeat study with PFMOAA at higher concentrations. And that's one of the things that's really important to remember for these compounds, is that we as humans excrete PFAS at a slower rate than experimental animals like mice. So we have to very carefully adjust our administered dose, or how much of the dose we give animals, so that their internal concentrations are going to be more similar to what people are experiencing. But for many of these compounds, we don't know what those internal concentrations are. So sometimes it's just kind of a touch and go in terms of how much you give and whether or not you're going to be able to determine if there's a response that might be similar to what humans are experiencing. So we've done 2 studies with PFMOAA. We've done 1 study each with PFMORPRA and PFMOBA. We've done 2 studies with Naphion byproduct 2, and we've done 2 studies with PFMOAA and a Naphion byproduct 2 mixed together. Our Naphion byproduct 2 paper is on my computer screen right here. My student has given me the final draft, so as soon as I edit it, we'll be ready to submit that. And she's working on data for our repeat PFMOAA study and for our mixture study. We've also now started working with some of the other compounds for our stage 2 studies, and should be generating those. Well, we are generating those data right now, so we're be analyzing those this summer. What I'm sharing with you now is a highlight from the paper that was just published in Toxics. And this is an overview of this TDAR, or the antigen-specific antibody response. This is analogous to the vaccine response. So what we have from A, B, and C is PFMOAA, PFMORPRA, and PFMOBA. We also included a PFOA positive control because we wanted to ask, are these compounds producing changes that are similar to what we've seen for PFOA? And if you look across these charts, you can see that there's no little stars for statistical significance. We didn't detect changes in this particular response for these compounds at the doses that we administered. We also didn't see changes in liver weight. We didn't see big changes in enzymes. We didn't see changes in body weight. We didn't see changes in the natural killer cell response, and we didn't see changes in numbers of immune cells. This doesn't mean that these compounds are safe or non-toxic. It just means at the doses we administered and for the endpoints we examined, we didn't detect effects. So I don't want you to think, oh, there's nothing to worry about, nor do I want you to think, oh my gosh, I should be terrified. It just means that at the doses we tested, we didn't uncover effects. Now for Naphion byproduct 2, and this is unpublished data that will be part of the paper that we're submitting hopefully within the next couple of weeks, we did see changes. And you'll also notice that our doses of Naphion byproduct 2 are lower than the doses than for the other 2 compounds. We use doses of 0.5, 5, and 50 for the C4 and the C5. Naphion byproduct 2 produced overt toxicity or greater toxicity. So we had to remove our 50 mg per kg dose group because after only 3 days of exposure, our mice were not doing well at all. And according to my timer, Jeff, I have 2 minutes left. So with Naphion byproduct 2, we did see suppression of this antigen-specific antibody response at that 5 mg per kg dose. We also saw increases in weights of the liver, increases in enzyme levels in the liver, changes in immune cells, and changes in natural killer cell cytotoxicity at that 5 mg per kg dose. We also detected levels of Naphion byproduct 2 in the blood at about 20,000 nanograms per mil at that 5 mg per kg administered dose. So what we've learned so far is that the shorter chain compounds, that is the ether compounds with 6 or fewer carbons, we think that they're fairly rapidly excreted from the body. We're still measuring some of these compounds in urine, but it looks as if at the doses we administered, they are eliminated from the mouse via urine fairly quickly. But that naphion byproduct 2, which is a longer compound with multiple oxygens, seems to be pretty toxic. It stays in the body, it builds up, it bioconcentrates, and did produce effects on the immune system, again, at the doses that we evaluated. So it seems that the number of carbon chains in the ether compounds also might be associated with accumulation as well as toxicity, which is similar to some of the legacy compounds. We still have a lot of work to do, and it takes a long time to evaluate these compounds one by one, but we hope to get a better handle, at least on this subclass of PFAS, so that these data might be used for purposes of better protecting environmental health. So thank you. And according to my clock, I have 47 seconds left.
Do you yield your time?
I do.
Thank you, Dr. Dewitt. And I think it's safe to say that without Dr. Dewitt and her team, there would be some of these compounds that we would not have any data for globally. So I appreciate her and her team making some strides in this. Not enough hours in the day or mice in the cage. So we've got a few minutes now for a Q&A. And I think I'm just like to go to the top and run down. We've got a question about pine trees. I will actually take this one because I talked to one of our air quality researchers about 12 months on this, and they were giving the analogy of a comb, that the needles on the pine tree, when the particular matter was in the air, would rest on the needles, but then when there was a rain event, they would wash off the needles. So I don't think the pine trees are actually magnifying anything with those compounds. I think it's just a pause of a rest stop until it rains, and then they come all the way to the ground. Dr. Bodnar, you're actually in the same department as the air team. I don't know if you want to weigh in on that as well.
Yeah, I think I would defer that question to Professor Jason Surratt. If you want to just send an email either directly to him or to the NC PFAS Network email, we can get that to him.
Okay.
I
There's
could
a question.
Jeff, if I could weigh in on that. There's actually some European data that shows that pine needles actually preferentially do absorb PFAS, and I'm working with another chemist, Aaron Baker at NC State. We have some data on PFAS in pine needles for the state of North Carolina that is coming towards publication very soon.
Interesting and ironic that it is now our state tree, or has been. There is a groundwater quality standards question about the practical quantitation limit of a 2 ppt limit for all PFAS. Would either Detlef or Lee like to take that question?
I can weigh in briefly, and then Detlef. I think it's that's one approach to take. The danger with using practical quantitation limits to regulate is that it relies entirely on the analytical technology, and it's a little decoupled from what we might know about effects or health impacts. So while I think it is important to make sure that we can measure these compounds at low levels so that we understand sources and fate, I think that it's probably worth considering the health effects when we're setting limits for these compounds.
Maybe I will add to this that the current North Carolina groundwater quality standard does mention the PQL, so it is written into our framework. So I do think that it's valid to then look at the PQLs of the methods. The challenge is that we have different PQLs or method reporting limits for individual PFASs in our methods, and also they vary from instrument to instrument. So I think for a standard US EPA method where these PQLs are well defined, I think that approach will work well, and I would support it. For other PFASs that are outside of a standard method right now, what I said earlier applies. It's going to vary from compound to compound, and it's going to vary from lab to lab. So it's a little challenging, but it does work, of course, for the standard EPA methods where these values are established.
So there have been a lot of questions on the Q&A board about biosolids. There's one up there still about earthworms and other soil-related organisms being analyzed for PFAS uptake in areas that have received biosolids. I know Dr. Belcher has worked on PFAS in alligators as an apex predator. I know Dr. Duckworth and Dr. Knappa have also worked with plant soil uptake. Does any of the three of you want to field that question on biosolids?
There's some data out there about earthworm uptake of PFAS from soils. Longer chains are taken up more readily than shorter chains, which is sort of what you'd expect. I don't think there's a tremendous biomagnification from soils, if my memory serves me correctly. I don't think anything that's being done currently. I don't know of anything currently being done in North Carolina on that.
Okay. Good question about Bladen Bluffs being the primary water source for Smithfield pork. I know we've looked at some plant uptake, but have we looked at any animal and food animals and water that's washing them down? Anybody from the NC State team? That one may just have to stay in the queue as we think about that, but that's a great question. Question about wastewater treatment plant operators being tested. I know Dr. Hoppen, Jane Hoppen at NC State is not with us tonight. She's been very active in this space with Dewitt and other NC State researchers and doing serology tests in the Cumberland County area. I don't know if a subset of wastewater treatment operators have been included in that. But Detlef, I don't know if you wanted to talk a little bit about your involvement with Jane and her work on the blood serology levels.
I was just trying to answer another question in the chat.
Sorry.
Can you repeat the question, Jeff? Sorry.
Well, there was a good question in here about have we focused on testing PFAS levels in wastewater treatment plant operators? And I was just talking about Dr. Hoppen's work with other NC State researchers in the serology tests. Didn't know if there was a general comment about what they've seen in the human population in the Cumberland County area.
So, and specifically on the question for the wastewater operators, I think that is an interesting one, and we could add to that workers at landfills as well. The first step in this, my group is involved in a Water Research Foundation project where we're actually looking at air emissions from wastewater treatment plant basins. My sense is that the exposure in the case of wastewater treatment plant operators would be through either volatilization of PFASs, and some neutral PFASs are volatile, as well as aerosolization of PFASs. And so, by looking at the levels of PFASs in the volatile and aerosol fractions, we can get a sense of, how important that exposure might be. In terms of, blood serum levels, we have published, through the Gen X exposure study that's being led by Jane Hoppen, PFAS levels in the blood serum of people in Wilmington, North Carolina, and what we have seen there is that some of the longer chain PFASs that were discharged into the Cape Fear River through the Chemours wastewater, accumulated in the blood serum of the residents in Wilmington. So for example, Naphthalene byproduct 2 was found in 99% of the samples that were tested. Also, we did find that the PFAS levels after 6 months in people who were tested twice, 6 months apart, started to decrease. And so that was at the time when also the PFAS levels in the drinking water decreased. So this showed that, you know, the improved water quality in the Wilmington area also led to a drop in PFAS levels in people's bodies.
All right, thank you
And,
for weighing
in
in on
terms
that.
of the Fayetteville blood samples, we're actually looking at these data right now, and so that is coming closer to you know, us sharing the data with the community.
Excellent. Well, thank you. Well, I tell you what, let's take a short break. It won't be a 10-minute break. Let's shoot for 3 to 5 minutes, and coming back around 7:10. And we will finish. We will go ahead and keep the unanswered questions in the live queue. Hopefully we can have time to answer those later. And I know some of our researchers are answering them directly with typed answers. So thank you for that. So we'll take a short break and be right back. All right, welcome back, everybody, to the second half of the PFAS Testing Network Public Symposium. Thank you for joining us. We have more than 100 participants tonight, and we appreciate everybody's time, especially at this late hour. We're going to go ahead and do a second Q&A after the next round of presentations, but we have a second poll for the attendees that we'll put up for about 30 seconds. So if you would be so kind, we love your participation. The poll should be coming up on your screen shortly. All right, great results from that. Thank you all for participating. Leading off our second round, I would like to turn it over to Dr. Duckworth, and he's going to talk about biogeochemistry. He is a soils professor at NC State. I think Dr. Belcher is also going to join in with him, who's a toxicologist over at NC State. So the floor is yours.
Thank you very much, Jeff. I want to talk to you about some work we've been doing to try to understand how plants that people might eat might take up PFAS from soils. This work was mainly conducted by two postdocs, Juan Bo and Cecile, as well as Professor Stephen Broom in my department, Dr. Knappe, who you've already met. And this is a joint project between Crop and Soil Sciences and Environmental Engineering at NC State. So what did we do here and what were we trying to do? So our goal here was really to understand how different PFAS might be taken up by plants and soil, and how we might be able to mitigate that by understanding how different components of soil might impact the transport of different PFAS into plants. So we started off with the whole alphabet soup here. We took a few different PFAS compounds. We took some legacy compounds of different chain length, PFBS being the shortest, PFOS being the longest, the ones we did. Some fluorotelomers of different chain lengths as well as POMO-AA and Gen X, which are fluoroethers of specific interest to the area around the Chemours plant. So what we did is a greenhouse study where we grew lettuce and we contaminated soil by spiking them with these compounds. And then that soil was mixed with clean commercial compost that we actually obtained from the NC State Farmers Market. And that compost was added at different rates to increase the organic matter content in the soil. So the idea being that if you amended soil, might it impact the ability of PFAS to be moved into the plants? We then grew the lettuce for the prescribed time, 45 days, in our greenhouse. You can see this lovely high-tech NC State facility here. Then we sampled and we did a bit of processing to get the PFAS in a form that we could, or the samples into a form that we could inject them into our ICP-MS MSs for analysis. We did this for the shoots, the edible part, the roots, also pore water that we extracted from these pots, and the soil itself to see how much had remained in there. I'm not going to talk about most of this data. It is useful for understanding how things transport through the chain from the soil to the plant, but mostly I'm going to talk about the shoots today because that I think is the part that'll be most interesting to people. So what I have here is a plot that shows shoot concentration in nanograms per gram of dry weight. So how much of each PFAS compound is in the leaves of the lettuce. And what I have here is different PFAS compounds on the on the x-axis. And also I have different compost rates here shown in different colors. So we have the soil unamended would be the white, and then the green would be 5%, 10%, and 20%. This amounts to an increase. I don't have the data shown here, but this amounts to an increase from going from maybe 0.5% organic matter in the soil up to about 4% organic matter in the soil. And what we can see in all cases, we see a fairly substantial drop in the PFAS that's transported from the soil to the plant leaves. So the compost is somewhat effective at lowering the uptake of PFAS into the plant and then its transport into the leaves. So we look at what's actually in the leaves, we can compare the different compounds themselves. So we did this all with one contaminated soil. And what we can see is the profile of what's in the leaves is really dominated by the short chain guy, PFBS, not the longer chain species, and also the fluoroether compounds. So we unfortunately see that JetX and PFMOA are pretty readily transported into the plant as well. Not quite as readily as PFBS, but more so than the fluorotelomers and the PFHXS and PFOS, the common legacy longer compounds. Okay, so what does all this mean? Well, we see that the GenX and PFOMOA are transported along with PFBS pretty well into the plant. And we also see that one of our primary goals of understanding that the adding compost dramatically reduces the uptake of PFAS by lettuce. So our idea was that potentially this could be a mechanism for maybe amending soils, or maybe if people want to use raised beds to grow produce in the region, maybe there would be some cushion or margin of error by adding high compost soils to this. But I think we still need more information. Everyone wants to know, is it safe to eat produce that's grown in gardens, in yards around the plant? And we don't have the answer to that because there's a lot to it, right? We don't know the toxicology of it. We don't know how PFAS is absorbed from foods into people particularly well, specifically these fluoroether compounds. But we definitely, and we also don't know about different plants. So we have some studies in progress. We have a freezer full of plants that we grew already that we're going to analyze for different crops. Including roots and seed parts and fruit plants. And we need more data on that to explain these things. But we also need to think about food in terms of overall exposure, right? So we all understand that if we drink contaminated water, we're being exposed to these compounds. How much is the more exposure results from food is something we really don't know. We can do some back of the hand calculations, but that just gets us in the ballpark. We need to get more information before we can really assess just how important the contribution of diet is to the exposure to PFAS. And in addition to just contaminated soil, we're starting to think about how contaminated irrigation water, potentially from wells that have been contaminated, and biosolids or even animal waste could potentially be transported from PFAS into agricultural products. Acknowledgements, want to thank some other folks who have contributed to it. And of course, the PFAS Testing Network and some facilities at NC State. And with that, I think I can maybe give back some time. I don't think anyone will complain.
All right, thank you very much. And I believe Dr. Belcher was going to come in now right before Dr. Lodfarth. And real quick, to whomever is rebroadcasting us over Facebook Live, thank you for that. We have been told that we've got folks joining us as far away as the Netherlands and Australia. So hello to our friends down under. And I used to play in a band with a drummer from the Netherlands who used to always say, hi, hi. So hi to our friends from across the pond. And now I will turn it over to Dr. Belcher.
Great, thank you. I don't know if I can follow Jeff's band stories, but I'll give it my best shot here. I'm telling you about one of the applied research teams. We're team 5B, and our project goal for the PFAS Testing Network was to really look into bioaccumulation and adverse effects of the PFAS. And this included Gen X and the fluoral ethers we've heard about, and to just generally understand how the mixture of the chemicals was impacting the health of Cape Fear River aquatic systems. We actually have published one paper so far and have a couple more in the works. Our first one was focusing on striped bass, really iconic species in North Carolina that is sitting near the top of the food chain. So we were curious to look at that for some of these bioaccumulation and the other, and I'm sorry I didn't turn on my video, I just realized here. The bioaccumulation and bioconcentration of these species. We're also using American alligators, and we're using these as real top of the food chain alligators that are long-lived. They reach sexual maturity in their mid-teens. They couldn't be in this water since before the 1980s. Some of these animals may be 40 or 50 or up to 60 years old. That we've been sampling from. So we really thought we had some good models that we could use here in the environment for some real health effects following real exposures. So our study goals were then just to use these guys as kind of that proverbial canary in a coal mine, to really look and see what kind of predictive effects we might be able to see in these animals that have had these chronic exposures that will vary. For example, in the case of striped bass, some of these animals may be moving in and out of the river system, but we were collecting them at times when they were up the river system and up in the Cape Fear during their annual spawn. And the alligators, they're actually a really nice species for these types of exposures because they typically don't move very far and, like I said, are long-lived. And we were looking at exposures, trying to define how many different PFAS we could see in their blood, and then looking at this question of bioaccumulation and bioconcentration and impacts on their health. We were really using quite a relatively simple hypothesis for this analysis. We had two watersheds that are neighboring that we knew had alligators in them. The Lumbar watershed, which we were planning to use as a comparator for lower dose exposure of the PFAS, as well as the Cape Fear watershed. And maybe we would, even see kind of a decrease in exposure as we went down this watershed was some of our initial hypotheses that really started this study. So it was really this canary in the coal mine concept that we can all understand of these indicators of health that may be going on similar to their human neighbors. And so what we did in the study, this is a picture of Teresa Gillette, a postdoc in my lab at the time, who's now moved on to the US EPA. We were collecting blood and collecting the serum fraction from this blood from both the fish and the alligators, I'll tell you about in a second. And we were working below Lock and Dam on the Cape Fear River. And there had been, over the years, there's been a lot invested in striped bass in North Carolina, especially in the Cape Fear River. There's annual stocking, and as it turns out, this species is non-reproductive in the Cape Fear when it used to be. And a lot of effort has been put into, take out the dams and allow the fish to be able to get back upstream. But really, that hasn't seen a lot of an improvement. So there's a big need to understand what is stopping these fish from reproducing in the Cape Fear River. So that was part of our idea. Our analysis of these PFAS showed that there was in fact very high levels of PFAS. And you'll note we've been talking about the water. The water is in parts per trillion or nanograms per liter. We're talking about nanograms per mil here in the blood. So really it's a matter of adding 3 zeros to the end of these numbers to get to those water things that you're familiar with. But what we did see that there was a lot of very high levels of PFAS in the fish from the Cape Fear River. Those fish were actually all stocked from the hatchery, so we knew their age. They were between 2 to 7 years of age. And most of the PFAS was PFAS. It accounted for about 70 to 80% in most of the fish. We compared these to fish in an aquaculture facility that had very low PFAS levels, just to demonstrate that we could compare those levels effectively. And really the big result of this, as far as the bioconcentration is in fact these chemicals, PFAS, Gen X, and Nafion byproduct, are all accumulating at higher levels than was in the surrounding water, suggesting very strongly that all of these compounds are bioaccumulating within the bodies of the animals. The other part of this that received some attention is really similar to the endpoints that Dr. Dewitt was looking at, that idea of looking at innate immunity, or the immediate defenses to bacteria and viruses and other nasties that we want to have that very quick response to. We looked at lysozyme as a biomarker and found that there was a association between increasing concentrations, here shown PFAS because it is the vast majority that is there, and an increase in this innate immune activity at times. So that was a very important endpoint that led us to really looking more deeply using the alligators. And again, I went over our hypothesis thinking that we'd have these changes in concentration here. Actually, as it turned out, we were right about this, that Lake Waccamaw had lower exposures than our animals associated with the Cape Fear River. But we also found something new that we're expanding our studies to look into, that we were able to see this other population based on their exposure to PFAS. And these are shown here. We call them coastal here, but in fact, all of these animals are living on golf courses. So we saw a very different signature in the animals on golf courses, even if they were on the Cape Fear River, than those ones that were associated with the Cape Fear River most specifically. Building off on our striped bass studies, we saw the same sort of linear increase with concentration, this association with this lysozyme activity. And in fact, here I've broken out our Lake Waccamaw alligators from our ones that were in the Cape Fear population. And you can see from here as we increase, and these are log units or going up, it's a log scale here, going up, increasing. We actually see that same linear increase in those lower levels in the Lake Waccamaw alligators compared to generally the alligators largely from Greenfield Lake and the surrounding waters on Cape Fear River. What we did see for health effects was a very interesting phenotype that's never been seen in any reptilian before. We are actually seeing this association with abnormal blood cells, which were only observed in the Cape Fear River animals. Here's one of them shown there. They're called schistocytes, or helmet cells. As you can imagine, it's kind of an upside down helmet, which led us to start to look in the idea of altered immunity, looking at autoimmunity. And we saw a number of different hallmarks of lupus-like autoimmunity in the alligators, showing you just really two things here. We have a number of other endpoints that we've looked at that support this, but this is double-stranded DNA antibodies that recognize DNA, which is one of the hallmark tests for lupus, as well as skin lesions and slow healing wounds on alligators, which is very rare at other sites, but was very typical of these Cape Fear River animals at different time. And alligators are known to heal very, very quickly. Typically, we've seen multiple animals that have lost a leg to another alligator, and those can heal up in a matter of weeks. But we saw these very atypical lesionous wounds. This alligator here is actually in the past had lost his tail to another alligator, but had these poorly healing wounds that were more recent. So we're really building this case of the evidence, really pointing towards immune deficiency, or not deficiency, excuse me, differences in these animals that may well be resulting in autoimmune-like diseases. So to summarize everything real quickly, PFAS do bioaccumulate in fish and the wildlife in North Carolina. There is evidence for harmful effects of exposure, including disruption of the immune system, which we're following up right now and hopefully going to have published very soon. One of the things that's really important to think about the PFAS Testing Network, which has been a real honor, is that science doesn't end. And we've leveraged these PFAS Testing Network studies for a number of additional studies, expanding these out. We're now doing annual monitoring of alligators and fish, and I'm going to leave it at that, including our community engagement work, working with environmental education, trying to get understanding of the PFAS out to the rest of the world. So thank you.
Thank you, Dr. Belcher, and appreciate that presentation. You'll note that there are multiple questions that have come across the Q&A board that you might want to weigh in on directly, either by typing or we can answer live at the end. I'm going to pivot now, and we're going to talk about mitigation measures. And I would like to introduce Dr. Frank Leibfarth, who is part of the Leibfarth and Orlando Cornell team. Orlando's over at Gillings Global School of Public Health and Environmental Science and Engineering. Dr. Leibfarth is with UNC-Chemistry. He's going to talk about some work he and his team have been doing in the lab on ionic fluorogel resins and their potential for PFAS removal from drinking water.
Thank you, Jeff, and it's an honor to be here and talk to all the constituents through the PFAS Testing Network. So, as Jeff said, I'm in the chemistry department at UNC-Chapel Hill, and I'll be presenting today on behalf of myself, my group, and also Professor Orlando Cornell's group, who is in the Environmental Science and Engineering department here at UNC, and has been a really close collaborator with this project. And here's my group, and I wanted to pick out Irene Manning, who's right there, who's done most of this work. She actually grew up in Pinehurst, North Carolina, went down to Florida for undergraduate, but now is getting her PhD at UNC-Chapel Hill. So she's really been the pioneer in a lot of this work. And here is Orlando Cornell and his team, who has been working on this project. So originally, when I got involved in this project, I didn't know much about PFAS, and it's been really great being part of the network and learning so much from all the fabulous scientists here. But when Jeff actually approached our department and I started thinking about this problem, right, the idea was to find a material that absorbs a lot of PFAS. And when I, as a polymer chemist, so my group traditionally thinks about how to make plastics, and especially how to make plastics more sustainable. When I think about polymers that absorb a lot of liquid, I think of diapers. So there's this class of polymers called super absorbent polymers that are found in diapers that, as you all know, keep babies dry, and they absorb a lot of water. So I thought about the design parameters that go into those super absorbent polymers and started to think about how can we incorporate those same design parameters into a material, but actually make that material selective for PFAS rather than for water, like these super absorbent polymers are in diapers. So when thinking about kind of the fundamental chemistry of how to do that, we came up with a hypothesis that included two specific things that we wanted to incorporate into this resin. And the key challenge we recognized initially is these PFAS molecules, right, are hazardous at very low concentrations. And typically there is non-fluorinated organic matter present in water at 3 to 6 orders of magnitude. So right, 1,000 to 100,000 times higher concentration than PFAS. So really the trick here is you have to pull a small amount of PFAS out of water that contains, much, much more non-fluorinated organic matter. So when designing this material, we wanted to take advantage of this concept called fluorophyllicity. Now, this has been known quite a long time and has actually been used quite a bit in the pharmaceutical industry to purify materials. This is essentially the concept that fluorinated molecules like other fluorinated molecules. They have a thermodynamic affinity towards those molecules. So you all know if you put oil and water in a glass, they will separate into 2 phases. If you put a fluorinated liquid into that same glass, it will actually create a 3rd phase. So fluorinated materials are neither hydrophilic or hydrophobic. They're what we call fluorophilic. So we wanted to couple that fluorophilicity with this concept of ion exchange. And ion exchange is, one of the preeminent ways that we currently purify water through ion exchange resins. And since many PFAS are negatively charged, we wanted to incorporate positive charges to get that electrostatic interaction in a purification material that would not only allow PFAS to go into that resin, but then would allow it to stick there. And we hope that these two concepts would, be more than additive. They would have a synergistic effect that would allow us to absorb a lot of PFAS. So the materials we started with are shown here. These are both commercially available, this Fluorolink material and an amine containing monomer to give us those positive charges. We were able to do a chemical reaction in order to make a cross-linked polymer network. So this effectively is a granular media. We can grind it into different size pellets and use that for purification. And then we can also go from this amine containing material to an ammonium, which is a permanent positive charge to really give us that electrostatic interaction. And so the plot I'm showing you here is 3 materials that are commercially available sorbents that are used for PFAS. And these 3 are particularly good at absorbing PFAS typically. So this is a variety of granular activated carbon and of a specific ion exchange resins that are typically known to be good at absorbing PFAS. The y-axis of this chart is percent removal of PFAS. So this is what's called a batch study, where we put a specific amount of this purification resin into a liter of water. We let it stir for 24 hours. Actually 21 hours we determined was plenty of time. And then we remove a sample of that water and see how much PFAS is left. So as you can see, granular activated carbon, for long chain PFAS like PFOA, adsorbed, about 50% of it in these specific water conditions. But as we know, the short chain PFAS typically are harder to remove from water. So things like the 6-fluorocarbon PFHXA and Gen X were removed less well. And I will note that this water contained, considerable amount of ionic content. So salt, 200 milligrams per liter salt, and about 20 milligrams per liter humic acid, and about 1 microgram per liter PFAS, which is an environmentally relevant on the high end amount of PFAS. So one of our optimal materials I show you here, and as you can see, it performs considerably better under the same conditions as these commercially available materials. So it absorbs, a large amount of PFOA and a significant amount even of these short chain PFAS, which was particularly notable. We then went on to perform batch experiments on real water samples. So this is water that that had contaminants that would mimic surface water, but we actually went and tested this on water we were able to acquire from the Wilmington Sweeney water treatment plant. And in this case, we test this on 21 different PFAS. And we can see in these batch studies that it absorbs a lot of these PFAS quite well, especially things like legacy PFAS, PFOA and PFOS. It's particularly good at absorbing. And for the shorter chain PFAS like PFBA, it is considerably challenging to remove those, but I will note that it's challenging to remove those for all purification technologies. And we did publish this paper and actually were able to submit cover art. And as you can see in this cover art for this journal, we were able to incorporate the state of North Carolina, the mountains in Western North Carolina, and the beaches in Eastern North Carolina. So that is what I call a proof of concept study. We showed that these design parameters, the fluorophilicity and ion exchange, did work to remove PFAS. But there were a number of challenges that we knew we had inherent to this initial resin that we tried. And one of the most critical to solve was the fact that the chemical bonds that connected this resin together were hydrolytically degradable. So in water over time, we hypothesized they would degrade. So when we were rethinking these resins, we made a second generation of resins, in this case where the chemical bonds that were connecting this network were not hydrolytically degradable. So these resins have a number of advantages, including this hydrolytic degradation. These resins actually performed even better in batch studies, and we plan to publish that data soon. But critically, we can see we did what are called expediated degradation tests. And we can see in our original material, in water, in these expedited degradation tests, we can see that, over time, 40% or more of these materials degrade, which is, unacceptable, certainly for a water purification technology, especially because, right, the connection points in these do contain fluorinated polymers. But in the case of our chemically stable materials, qualitatively, these are much, much more stable. And we are going on to do more quantitative studies with non-targeted analysis to really make sure these don't degrade. And lastly, I will say we now are looking to scale up these materials to larger scale to actually try, at scales that would be relevant to a wastewater treatment plant or at an industrial facility with large volumes of water. And we've now identified these Lumiflon type materials that we can make these resins from. And these materials have a really high hydrolytic stability. They're very inexpensive. They're actually a primary component of outdoor paint. And we're now using kind of this as a basis for the new resins we're making. And we are doing a lot of now packed bed resin studies where we're flowing water over a resin. And again, finding that in these contexts, this concept of fluorophyllicity and ion exchange is really providing benefits over current state-of-the-art technology.
All right, Dr. Lodfar, thank you very much for that. We've got some more Q&A. We'll get to that in just a second. I would like to thank a few groups of people. First of all, I would like to thank our legislators who actually reached out and suggested we do this forum as an update after turning in our final report, which is available on our website, both at the Collaboratory, which is collaboratory.unc.edu, as well as the PFAS Testing Network. I would also specifically like to thank our research assistant, Hope Thompson, who has done a lot of logistical work in the background, getting everybody's schedule lined up, getting all the presentations, setting up the logistics on Zoom, handling our chat and our Q&A in the background on Zoom. We could not have done this without Hope. And Hope is just about a week away from earning her master's in public health at Gillings Global School of Public Health. So what an academic year it's been, but what a way to go out with a bang. So thank you, Hope. Thank you to all of our researchers. We could not have done this without you. As Lee Ferguson likes to say, it's unfortunate that the PFAS issue happened in North Carolina, although it's happening all over the US. But if it were to happen in a state, this was a great state for it to happen in, because we have such an amazing concentration of PFAS-centric researchers. And you've just seen a small portion of the breadth of work that's been done over the past few years, and that will be ongoing, and we're hoping to continue this work into the future. So what would be great is if there are any questions. We've got one up on the board. Are there plans to study PFAS in the estuaries or in what about oysters, shrimp, crabs? Scott Belcher, you want to weigh in on that? By looking beyond striped bass and alligators?
I was typing as furiously as I could to answer that, but I think there's a pilot study in the CHHE, I believe, that is looking at either mussels or oysters. I'm not 100% sure, and we have begun and we are about to look at least 1 or 2 blue crab samples that I have in the freezer, developing a method for that. And some other, funding dependent possibilities include some offshore fishes also.
Okay. Does anybody want to weigh in on some general comments about the EPA? Obviously, if you wanted PFAS to be on the radar of the EPA, then Michael Regan heading up as the new administrator is a wonderful thing, because he's certainly very familiar with the challenges that he faced as Secretary of DEQ in our state. So do any of our researchers want to weigh in on the current efforts or potential future efforts of the EPA? And I saw something before, I think the question's been answered, about 29 compounds. I believe we've been target testing, what, over 40 at this point?
48, I think is the number, yeah.
48. So, but does anybody have any general comments about EPA and working with them? I know we've got a tight working relationship with Mark Striner and Eddie Lindstrom here in RTP.
I think I would just note the question, I think that, or the comment that was asked is basically about the final report suggesting plans or assisting the state in developing an MCL. I think, if anything, I mean, the amount of data that we've generated for water supplies across the state, I think that's all going to be available for the state and for the federal agencies to use as they develop those MCLs. So for sure, even though we didn't necessarily specifically call it out in the final report, the data is absolutely going to be essential, I think, for developing those MCLs.
And I'd also like to add that I think some agencies are going to have to start making some different approaches when it comes to classes of chemicals like PFAS. There are almost 10,000 individual substances. I can't possibly evaluate the toxicity of each and every one of those. And with something that's as persistent as most PFAS, perhaps we don't need to wait for toxicological data to make a management decision. Perhaps we can use their persistence and their potential to bioaccumulate and what we know about the well-studied PFAS toxicity to make some regulatory or at least basic management decisions.
And I've seen some regulatory questions. Certainly there's a role both for the executive branch as well as the legislative branch here. I believe I saw the statistic on the Twitters earlier today from Clean Cape Fears account. And I believe it was that the General Assembly has now filed in either the House or the Senate more PFAS related bills than ever before. So that's a great thing. Crossover date is coming up in the next few weeks when it has to cross from one chamber to the other. It will be exciting to watch. We don't know yet if any of the recommendations from the final report that came from each of the teams will be incorporated, but we'll certainly be watching for that. And our researchers are always available to legislators or state agency officials that want to reach out and are well known in those spheres and utilized a lot. So keep your eyes on the General Assembly and also DEQ, because they also have some regulatory authority. So are there any closing comments or any on the remaining Q&A board that anybody wants to talk about? Can't take the bait on that. Okay. So I did see something about stuck in rules. At least I've known that rules now becomes the passage point before they get re-referred to other committees. I think it was years ago that it used to go to Ways and Means and then you knew it was dead. So hopefully the rules is just a brief rest stop, but again, only time will tell. I think everybody is monitoring that activity and we're hoping for continued research. As I say, we've got a tremendous team of researchers that have spent some time here. I wish you could have heard from them all. There are so many more opportunities, but North Carolina has come a long way in the past few years. Of course, with data, you need certain data to make regulatory decisions. We hope that there are enough out there for our legislators and our state regulators to start making more decisions with those. Of course, it also opens up more questions, which begets more research. But we have come a long way in the past 3 years in this state. And you're looking at some of the world's leading researchers on your screen right now. And there are many more that are not present. So I want to thank them. I want to thank you all for tuning in. I want to thank our friends outside of North Carolina and outside of the United States as well. I do know this has been recorded. I do hope this will be up on one of the websites so that it can be replayed later for others that can watch it asynchronously. If you have any other questions that maybe didn't get answered tonight, certainly you can reach out to the individual researchers, but to also make it easy, you can email collaboratory@unc.edu. I'm going to put that up in the chat just because some folks, Collaboratory can be a challenge sometimes to spell. because it's kind of a real word and it's kind of not a real word. But that comes into us, and we will get those out to the researchers we think, or multiple researchers, so they can answer directly. So thank you. It's been quite a year, so I hope that everybody is safe and doing well and healthy, and please stay that way. And thank you for joining us, and have a great evening. And we might have another poll. I don't know if Hope's going to throw one up or not, but we have time for that if she wants to. So if you get a chance, we'd love for you, before you log off, to vote in this poll and let us see what our participant interests are. And when this poll closes, you may also have a survey launch. It would be great if you could participate in that closing survey. All of that feedback and information helps us learn from this and deliver a better product the next time we do a symposium. All right. Water and health effects leading the way for sure. Well, again, thank you to all our panelists. Thank you to our legislators that joined us in the beginning. Thank you to our attendees, and have a great evening.